alignment, we can see the sequences that is aligned, and we can see the where is the change. So PAM use of the PAM matrix is one of the example. So we will discuss in the detail also. And the fourth one is the diagnosis in the individual at risk. If if the people are suffering from the genetic disorder or some of the disease, we can diagnose those individuals. We can take out the sequences from those individuals and we can identify. We can sequence them and we can analyze those sequences. So analysis of the sequence variation has application in field other than the human disease. So this thing is also applicable in other fields, like in the crop improvement. Okay, and we can also study. We are all also interested to know about the origin of the human. I mean, like phylogenetic analysis. So in that, the variations are also important. So now today we will discuss about the some definitions. Okay. So there were the what we have discussed on the last term. Now at this stage, we will be able to understand what are the different definitions related to the sequences. Because in the last class, we have discussed about the phenotypic variations. Okay, that is visible, that we can see by our eyes. We can observe that in the nature, and that we have understood using several examples like the Negroes population, Caucasian population. Okay, and we have the diverse variety of plants and animals on the earth, so we can easily observe the differences. So that is all due to the genetic variation. So as the Mendel discussed, Mendel is the father of the genetics. So Mendel discussed about all these things. So he took the varieties of pea and crossed to get the hybrid varieties or pure varieties. So Mendel has described that there is some one element that is responsible for the variation of the characters. So although at that time Mendel was not aware that this is the DNA molecule and what is DNA, because discovery of the DNA was have not been discovered at that time. Okay. So, but the Mendel has given one term that is called elementy. So elementy is used. There is some element, but Mendel has already told us that there is some element, there is some chemical that is responsible for. There are some elements in each individual, in each organism that is responsible for the genetic variation. So that was the important clue. So later on, the Hugo de Vries and other scientists have worked on that, and they have identified the mutations. Okay, there are some other scientists also. I mean, it's like Darwin and Lamarck. They have also worked on that and provided the important theory of the evolution. So evolution is based on the variation. So now today we will discuss these definitions. We'll try to understand what is polymorphism. So now you have understood means all the information of any individual, of making plants, of making frogs, of making animals, that is all saved at the DNA level. Okay. So in 1944, the molecule has been identified. That is the DNA molecule that is responsible for the variation of DNA, variation of the phenotypic features. So there is one molecule that has been identified. Before that, there was an important experiment also that is given by the Griffith. Griffith has also told us there are some element, there is something that transfer the character from one generation to another. So Griffith has done the important discovery, but in 1944 the molecules has been identified. Okay, So we will discuss about this. We can discuss about the detail. I means what they have done about the important discoveries in the another class. So today, just we will try to understand what are the definitions. Means what is this polymorphism, sequence polymorphism, single nucleotide polymorphism, other term SNP and CSNP. What it means actually, we know that this is the DNA molecule. DNA molecule is responsible for the variation that appear in the next generation because DNA has the code or information that is needed to construct the organism. So the our genome, what we say that is our genome. So genome we all organisms have the different genome, genomic diversity we have. So our genome is responsible. So in the genome we have the different gene sequences and that shows the polymorphism. What is polymorphism? Sequence, what is sequence? It is DNA. Obviously, it is DNA. We are talking about the DNA because DNA sequence has the code to construct the whole organism. Sequence polymorphism. A sequence polymorphism is variation in a DNA sequence that occur at a frequency of greater than 1% in population. Because if the percentage is high, more than 1, then we are able to identify that variation in the population. What is the difference
हेलो
between
मैम.
Caucasian
हेलो
and Negroes? Okay, yes. What do you want to say?
मैम. मैम बाइलिंगुअल में पढ़ाइए मैम, थोड़ा सा कम समझ में आ रहा है.
What did you say?
मैम हिंदी और इंग्लिश को मिक्स करके पढ़ाइएगा मैम, कम समझ में आ रहा है थोड़ा सा.
Okay, in the last on the last turn you have understood.
यस मैम.
Okay, so I'm not using very tough word. I'm using the simple words. But you should listen. You should try to understand what I'm saying. That is not a difficult language. If you can understand in Hindi, you can also understand in English also. That is not a tough task. Variation, do you know that is variation is a change and that is observed at the phenotypic level means by the appearance you can observe means this is a plant, mango plant, apple plant. You can see the difference in the appearance. So that are variation. Now we are going to understand this at the sequence level. Okay, if the changes are more than 1% in the population, we can observe that at the phenotype. What is phenotype? Means that is the English word, so that because all of you understand that. Okay, in the genetics also you have read about these terms. Although I can take the another lecture that will also give you the basic information. Okay. Just I will try to make another lecture that will include the simple basic things. So that will give you more understanding about this. Okay, but to go along with the class, you should be familiar with this language, the English language, because everywhere you need this. You know, you need to improve your skills also, communication skills. If you will not improve now, then you are going to face the problem in the future also. So that's why I'm taking the class in English. Okay, because this is the requirement of your field. Functional polymorphism define variant that does have an associated phenotype. So what do you mean by functional polymorphism? Means it has impact on the phenotypic level or we can observe that variant function differential. Means it performs the means there was a two variety of like dwarf and tallness. Okay, so that was the function. And there may be some proteins, genes and proteins that may be responsible for that prediction of those variety. So there are certain variants that have and associated phenotype. Okay, so that is what the sequence polymorph. We need to study the polymorphisms at the DNA level. If there are the different variety of the same plant, like the Mandel took the different varieties of pea plant. So I'm taking the example of the Mandel experiment because you are familiar with that. So in that case, we have the different varieties. What is the functional polymorphism? Like tallness variety, tall variety, dwarf variety. So that was the two. In that, we have the tallness genes also in both of the varieties, but it has become recessive in one, and dwarfness genes also. So in some of the population, it has become recessive. So associated phenotype means if there is a phenotypic variation that is associated with that gene, like tallness. So there was one character that is the height, and there were two traits of the same character. Height means tallness and dwarfness. So that is the phenotype. That is all about the phenotype. In the phenotype, we can have the different trait of the same character that we can observe, like in the Drosophila, eyes color. Phenotype means we can observe, and gene we cannot observe at the I mean until or unless we don't have the sequence, and we can do the multiple sequence alignment. Then only we can know what is the difference, what is the mutation. By the alignment, we can analyze. Using the alignment, we can analyze the variation at the DNA sequence level. If you're talking about the sequence polymorphism, that means it is at the sequence level. So using the sequence alignment or multiple sequence alignment, we can show this is the mutation. These are the changes. You can compare the same protein sequence or same DNA sequence from the different organism to show the difference, like the hemoglobin from different organism, like mouse, human. So you can compare the hemoglobin of mouse with hemoglobin of human. But there is no phenotypic difference, I think, observable. But if you compare the sickle cell anemia, I mean the person who has the sickle cell anemia, so that is defective hemoglobin, and one person has a normal hemoglobin. So there is a phenotypic difference. Phenotypic difference means it is not observed at the body level, but it can be observed at the structure of using the structure of RBCs. RBC has a sickle shape. So that I mean you can say there is the change in the structure of the RBC. The person who has the sickle shaped RBCs, so that person is not be able to carry out the more oxygen. Means he will feel the difficulty in the respiration because oxygen carrying capacity of that type of RBCs is less than the normal RBCs. So, what is sequence polymorphism? This is the variation at the DNA sequence. So, at least we should have the DNA sequence. So, at this time, we have the DNA sequence of so many organisms. We have the genome sequences of multiple organisms. We have the database. And from the NCBI, we can retrieve those DNA sequences. And we can compare those sequences. And we can also detect the changes. What are the changes? We can compare the sequences that have been extracted from the different types of the population. You just go to the database, NCBI database, and you can retrieve the genome sequences. Then you can make the comparison using BLAST and FASTA. So using the FASTA is the tool that can be helpful here. Okay, it uses the concept of pairwise alignment, but genome comparison is there. You can perform that. Anybody has the doubt at this point? Sequence polymorphism. If anybody want to ask the question, can ask. Do you have any question about the sequence polymorphism? Now there is a difference between a sequence polymorphism and functional polymorphism. For functional polymorphism, means you are able to identify the effect of that change in polymorphism. Anemia. So in case of sickle cell anemia, you are identifying the effect of the mutation sequence polymorphism at the functional level. Also means what is the structure difference between the RBCs of the normal and abnormal. So this is called the functional polymorphism. If we are detecting the changes at the protein level also, or at the expression level, is what is being shown in the expression. There is another definition, that is single nucleotide polymorphism. If the sequence has only the single change, like that was also the case of RBC's sickle cell anemia, there was only single change. So what is the difference between SNP and sequence polymorphism? In sequence polymorphism, we can have the multiple changes. If we are saying that there are two different populations, so there must be changes that is more than 1% in the population, then only you can say so these are the different population. So you can easily observe the difference, like between the plants, variety of rats, white rats, brown rats. So these are the two varieties, because we observe the change at the phenotype level, means they show the different skin colors. So their population may be different, like the Negroes population are peoples that are fair in the color, because you can identify the difference that is observed, like skin color, black or white, black or fair. So if you are saying that the sequence polymorphism, that is at the population level, but in the SNP, that means we are talking about single change. That is a case of sickle cell anemia. If we are discussing the sequence polymorphism at the population level, so that is the population, I mean sequence polymorphism related to the population, means large number of individuals. And there is a difference. By looking at the individual, you can identify, like you can compare the Manipuri people from the North Indian people. There is a difference among the appearance features. So in the sickle cell anemia, you will not be able to identify that SNP variation by looking at those individuals. Okay. The person who carried out the blood with the defective RBCs, you will not be able to identify that. You need to carry out some experiment. Then only you can identify, or there will be some diagnosis. This persons feel difficulty but by looking at those individuals, you cannot identify. But there will be certain symptoms also that you can identify the effect of SNP. So this is the difference. SNP is a polymorphy in which alleles are defined by single or few base changes in the human genome. So these comprise mainly substitution, but the term also embraces deletion and insertion of one or few bases. Okay. So this has very limited changes, single base changes. So it can be insertion, deletion, or substitution. Okay. So here we are discussing about the two types of the polymorphism, that is sequence polymorphism and a single nucleotide polymorphism. So there is a difference between these two types of variation. Sequence polymorphism, that means it is related to the population, whole population. So we can have the two types of population, and we can observe the changes among those population. In the previous classes, I have told you about one example like thiopurine is one drug. So that is metabolized in the presence of TPMT gene. But the TPMT gene is absent in the Caucasian population. So that population is not, so this drug causes the toxicity in the Caucasian population. Because those Caucasian population, individuals of those population, that population lack that enzyme, TPMT, thiopurine methyl transferase enzyme. So that is required to metabolize that drug. So that is a functional polymorphism. Means we can observe the difference. So all the individuals of that population, Caucasian population, won't have that enzyme. So that is one example of sequence polymorphism. And that is at the population level. Have you understood this? What is sequence polymorphism is now clear. That means we are talking about the population level, the large scale. If we are saying the frequency of 1%, frequency should be greater than 1% in the population, that means we are analyzing the so many individuals, and that should be 1% in the population. Then the DNA sequences have also been analyzed, and the protein sequence also we can also analyze, because the protein gives the clue about the function of them. TPMT is absent in the Caucasian population. So this will tell about the function. So that is also, so here we are also discussing about functional polymorphism. Means function is important. Function of the enzyme is important. If the enzyme is defective, if enzyme is not present there, or that may be inactive. So define the variant and does have an associated phenotype. So what are the associated, if we are studying the associated phenotype of that DNA sequence, then we call that functional polymorphism. Means we are going to study the effect of sequence polymorphism on function at functional level. That is functional polymorphism. Functional polymorphism shows the effect of the DNA sequence polymorphism at a protein level that you can observe. And you can identify this using the experiment also. And by applying the drug, you can give them the drug, so it will cause the toxicity in those individuals. Because the person, they will not be able to metabolize this drug. So that is the example of sequence polymorphism. A single nucleotide polymorphism is the change that occur at in the allele that is defined by the single and few base changes. So this comprise mainly the substitution, ingestion, and deletion. So we'll also study about the mutation, about changes at the phenotype level. So mainly our focus is the SNP. SNP has a great role in the medical field also, because we are interested to know about the genetic disorders that is occurred in the human. So these are the different terms like ASNP and CSNP. So they describe the anonymous SNPs and coding SNPs. So anonymous SNPs are present throughout the genome. These are dispersed. These are not associated with some particular position. And that is not associated with some specific coding sequence. So that are called anonymous SNP. So that can be that can be found in the various individuals of this. Uh, I mean, in the different population also, and in the same population also. So there may be the variant, but we cannot fix the position. The position is anonymous, means it is dispersed, that is found throughout the genome. So that is called anonymous as single nucleotide polymorphism. Is this clear? Dispersed throughout the genome. ASNPs,
Yes, ma'am.
that is dispersed throughout the genome. So that is anonymous. We are not giving the name here because it can be found anywhere in the genome. Okay. And there is another type of CSNPs. That means C means coding. A means anonymous SNPs and C SNPs means coding SNPs. Okay. Coding single nucleotide polymorphism. Coding SNPs. What do you mean by this? It is, I mean, by this term you can understand. Means coding means it is affecting the coding region. SNP is single nucleotide polymorphism that are found in coding region. What is the coding region? Means coding region of the gene. Obviously, it is gene exon region. So these coding SNPs played an important role. That is important to us because it will affect the production of the protein. Obviously, there will be a change at the function level. It can change the function of the protein. So that is important to us. So we are always interested to study about these coding SNPs because this has the medical importance also. Nowadays we are getting the different kind of genetic disorders. So because in the human population we are getting the disease associated with that those disorders. So the coding SNPs is the case. Coding SNPs is associated with those type of SNPs. Okay, so these are the two. So here I want to say, if you want to study more about this, means there is a, if you want to do some practical related to this, you can go to the NCBI databases. From the pull down menu, you can select the DBSNP. There is a database that is called Single Nucleotide Polymorphism Database, DBSNP. So there we can study about these SNPs. We will study in the next lectures, coming lectures. I will take, I will show you some practical exercise also. Okay. I will show you how it looks, how we can retrieve that type of information. Now, if you have any doubt here, you can ask. So I think these concepts should be clear to you. Okay. I have described you. There are the two types of polymorphism, that is sequence polymorphism, and another one is the single nucleotide polymorphism. So first one is associated with the population, variation among the population, and this is the second single nucleotide polymorphism in which the allele shows the difference by one base or single base. Okay, few base changes in the human genome. Okay, so it can include the substitution, ionization, deletion, and there are the some two types of the SNPs, that is anonymous and coding SNPs. Okay, now move to the next topic, mutation, because obviously SNP is also mutation. But what is the difference? Mutation by contrast with the polymorphism. A mutation is change in sequence that is directly associated with the change in phenotype. So what is the difference between SNP? I mean, like the ASNP, anonymous SNP, SNP is detected as a multiple sequence alignment. If we have two sequence, mouse and human, it shows the SNP. But we don't know about the effect of that mutation on phenotype. So that can be SNP. Means we don't know about the effect. That effect is not observed actually. So that is the SNP. But mutation, what we say the mutation? So this is the polymorphism which may not show the effect at the phenotypic level. But when we say this is the mutation, mutation is a change in the sequence that is directly associated with the change in phenotype. So here it is defined like this. Although we study, I mean, we call all the changes, we call them the mutation. In the books also it is written the any change at the DNA, that is the mutation. It show the effect at the phenotypic level or not. We call them the mutation. Generally, we call them the mutation. But here it is emphasized here that we should not call them. So mutation is a change in sequence that is directly associated with a change in phenotype. So when it shows the change at the phenotype, it becomes important. So that mutation become popular because that is of our great interest, because it shows the effect, like the mutation in case of sickle cell anemia. So that is important to us because that is affecting our individuals, human individuals. Okay, so that is the difference. I mean, so they have shown, they have defined the mutation in the another term to differentiate. That is only to differentiate from SNP. Okay, to differentiate from the polymorphism. So they have defined the mutation like this. Genotyping. Genotyping is the analysis of a locus to look for the presence of known allele. Now, what is genotyping? That means we are analyzing the locus also. What is the location of gene to look for the presence of known alleles? So that genotyping, Mendel has also done that. I mean that we can do by the observation, by looking at the seeds, the shape of the seeds. Mendel took the character like shape and height, shape and color, like yellow and green, and round and wrinkled.
एक्सक्यूज मी मैम।
Yes.
मैम नेक्स्ट क्लास का टाइम हो रहा है, अटेंडेंस ले लीजिए।
Okay, I'm just closing this. This is the last slide that you are studying. The other thing we will study on the next turn, but I want to complete this. Okay, so genotyping is the analysis of locus to look for the presence of known alleles. So this we can, how we can identify the location of the gene? That is called gene mapping. And gene mapping can be done in the physical way, physical mapping, genetic mapping. Physical, what is physical mapping? That is the identification of locus of the gene at the sequence. So we are identifying the location of gene. We are identifying the distance between the two genes at the sequence level, physical distance. We are detecting the physical distance. So that is called physical mapping. In physical mapping, we use the genome sequencing methods. like the top down approach and bottom up approach that we have discussed in the initial classes. Top down and bottom up approach of DNA sequencing. Can you remember which method is important for gene mapping? I have already told you this in the class. Which method is important for? One method is top down approach and another method is the bottom up approach. So in the top down approach, we have the global view of the sequence. We start from the top and we start from the whole genome, then we divide the whole genome into the multiple segments that are called quantics. So that method is important for the gene mapping, that is used in the gene mapping. So we use that. We have the all the schematic view of the all the overview of that whole genome, and we can identify the location of the gene. And in the bottom up approach, we also identify the location of, if we are only interested in the specific gene, in the presence of a specific gene.
मैम
Yes. Yes.
मैम नेक्स्ट क्लास का लिंक आ गया है.
Okay, but now it is 2 o'clock. Now you have told me that there will be class at 2:15. What is this problem? I had already wasted the time. I was here at 1 o'clock.
Yes, ma'am.
Okay. Now just wait for. I'm just closing, and this is not the way. Yeah. You only told me that he will take the class at. I mean, sir will take the class at 2:15. Just don't disturb me. And besides, we don't want to repeat it again, again and again. The last slide. So this is the genotyping, is the analysis of the locus to look for the presence of the non-allele. So you should fix up the times. I don't take the class in others' timings. You shouldn't disturb others. I have so many things to do. Since at 1 o'clock I was waiting for you, but you didn't join, and you did not also confirm. So mutation and genotyping, this is the thing. Now you know the difference in the mutation, genotyping. So physical mapping, genetic mapping is also important. Physical mapping, I have already told you, top down and bottom up approach. And one method is also left, that is called genome mapping. In the genetic mapping is done without doing any physical sequencing, without any genome sequencing. That is on the basis of the like Mendel cross or crossing with the breeds, different types of varieties, crossing the different types of population. So we can observe the changes among the offsprings. So by analyzing the total number of recombinase, like what are the recombinase, and the population can be divided. Recombination frequency can be calculated, and that recombination frequency is used to map the location of the gene on the whole genome. So there we take the assuming distance, that is called Santi-Morgan. That is it is named by the name of the scientist, Santi-Morgan. Morgan has told about that. So Santi-Morgan is the assuming distance, that is the overall distance. So we assume that recombination frequency is used as a Santi-Morgan distance. By considering the whole genome as a means total 100%, so we use the percentage. Like if recombination frequency is 5%, the distance is the 5 Santi-Morgan. So that is how we always interested to know about the locus of the gene, and we can use that. What are the different in the analysis of the different kind of population. So without doing any experiment, genome sequencing experiment, like from the Mendel population, we can identify the location of the gene using the only recombination frequency. So this is the genetic method. Genetic mapping is a method that is based on linkage and crossing over phenomena, and like pure hybrids are pure population is produced, as well as recombinants are also produced. Okay, so on the next time we will discuss about this. Now just type your attendance. I'm not going to take your attendance. Just type your roll number, name and roll number in the chat box. Okay? Type your roll number and name in the chat box. Just do it fast. Just type your name and roll number. Roll number also? Okay. So that this is your attendance for this class. Do it fast if you want to attend another class. I think it will be at 2:15, no? The sir will take class at the 2:50, no? So why you were disturbing me at that time? So that actually deviate me. Type your attendance in the chat box. Give your attendance, then you can leave. Type your attendance in the chat box. I'm again recalling, just type your attendance in the chat box so that nobody is left. Type your attendance in the chat box, then leave the class. PPPs are left. Just type your attendance in the chat box. Type your attendance in a message. There is a message icon on the right hand side. At the bottom, you just see on the right hand side. Okay, some of the students are not able to see. Somebody has called me regarding that. Just see at the bottom on the right hand side where 7 is written. Now everybody has done that. Everybody has given. I think nobody is left now. So I'm leaving the class now. Okay. Okay, two are left. Sidra is asked. Just give your attendance. Type your attendance in the box, in the message box. Then you can leave the meeting. After Saurabh, just do it fast. Nandini, give your attendance. Nandini, type your attendance. Okay, it has been done now. So now you can leave.